Hi Dr. Song, I'm Julie. The title of my presentation is GWAS and other multi-omic-based approaches in combating COVID-19. First, COVID-19 is a pandemic infectious disease caused by SARS-CoV-2 which belongs to genera beta coronavirus, and this virus has 6 ORFs. The accessory protein of the SARS-CoV-2 is unique in terms of the number, genomic organization, sequence, and also the functions. SARS-CoV-2 is an enveloped virus covered by spike proteins containing plus single-stranded RNA within nucleocapsid protein. These spike proteins have different types of the receptor binding domain which bind to angiotensin-converting enzyme 2 receptors to get entry into the host cells. Mutated RBD of SARS-CoV-2 is closely related to virus sample from bat, thus believed that this virus is a zoonotic pathogen originating from bat. Mutated RBD also has higher affinity to ACE2 receptor in human. The type of inheritance patterns of COVID-19 is X-linked dominant because ACE2 is X-linked gene. Thus, men have higher prevalence and severity of COVID-19 than women. These statistics show that more men die of COVID-19 than women. Besides genders, higher ages, cardiovascular disease, and BME communities also have higher susceptibility to COVID-19, including the blood group A, but blood group O has lower risk of susceptibility to COVID-19. Next, I would like to talk about the GWAS procedures, which include 6 steps. The first step is the sample collections and phenotype determinations. Case subjects are severe COVID-19 patients with severe respiratory failure which can be recruited from the hospital. They will be given the RNA-PCR test to confirm the diagnosis of COVID-19. Control subjects are healthy volunteers and blood donors selected randomly. To extract DNA, whole blood sample or Buffy coat will be collected through the venipuncture technique. The second step is the ethics committee approval, which is necessary before conducting the research. Informed consent must be signed by all participants to ensure enrollment, and this informed consent given is depend on the local ethics committee's recommendations and special policy issues for COVID-19 research. The step 3 is the sample processing and genotyping. DNA extraction from whole blood samples or Buffy coat using this machine with the low volume kit or Buffy coat kit. Then magnetic beams method is used to extract DNA. Illumina platform is also used for genotyping. This is the brief procedure of genotyping from the quantification to amplifications, followed by a series of processes, lastly imaging by the infinitum LCG scan to read out DNA. Initial genotype calling uses the Illumina software to extract GSA genotype data from intensity data file. The data is then used to remove samples which are less than 90% correct using Flink. The fourth step is the sample quality control. To qualify the sample, variance of samples with more than 2% of missing data, increase or decrease in heterozygosity rate, MAF less than 0.1%, different missing genotype in case and control sample or deviation from the Hardy-Weinberg equilibriums need to be removed. For relatedness testing, LD-prone success of SMP will be conducted, and also the principal component analysis, which used to identify the ancestry outliers using LD-prone success of SMP will also be conducted. Population stratification need to remove to prevent the inflections of the type 1 error with the remaining spurious population structures. And these tools are the example of the principal component analysis. The step 5 is the genotype imputations. After quality control and filtrating SMP with alleles 80 or GC, genotype imputation for chromosomes 1 to 22 and X using the TopMat FreeSpy and the Michigan imputation server will be conducted. The last step is the statistical analysis. Two analyses, which are the main analysis and analysis corrected for age and sex, will be conducted. Fixed effects meta-analysis will also be conducted by using the meta-analysis tooth method, quantile plot, and the Manhattan plot. The upper is the quantile plot and the lower is the Manhattan plot. Common acceptable thresholds of the meta-analysis need to follow in order to determine statistical significance. Next, Bayesian file mapping analysis will also be conducted, and the data is then inserted into the file map with the data from meta-analysis. 3 ABO SMP will be selected to infer ABO blood type and calculate odds ratios based on data from top-met genotype imputations. Then I would like to discuss about the results. There are two loci which are G variant at locus 3p2131 and C SMP at locus 9q342 with 95% of confidentials interval are possibly associated with severe respiratory failure caused by COVID-19. The association signal found at locus 3p2131 comprised of 6 genes as shown in this figure. The risk allele of this locus is associated with decreased expression of CXCR6, increased expression of SLC6A20, and strongly expression LZTF-L1 in human lung, which is possibly increase the susceptibility to viral entry and infections, thus showing that these genes are relevant to COVID-19 susceptibility. Patients who receive mechanical ventilator supports have higher frequency of risk allele, and the patients with homozygous risk alleles are found to be younger than the patients with heterozygous or homozygous for non-risk allele. The association signal at locus 9q342 is corresponding to the ABO blood group locus. Blood group A has higher risk, but blood group O has lower risk to the COVID-19 can be due to the presence of the A or B antibodies in the plasma, allow it to identify the foreign protein on viral surface better. The possible limitation of this research is the information about the infection status of control participants is limited because some can be asymptomatic. Thus, this can increase the risk of infections to both participants and researchers. The solution to overcome this problem is providing all control participants with the diagnosis test of COVID-19, and it is advisable to conduct at least twice time. Molecular tests and antibiotic tests are the example of the diagnosis test for COVID-19. The possible limitations for this research is information about the COVID-19 is limited and also need to recruit large amount of participants for this massive study in a short period of time, which cause only bare clinical metadata can be requested, thus cannot elaborate genotype phenotype of current findings extensively, and also cannot adjust the potential source of bias. The solution to overcome this problem is use the data from biobank or national dataset, which can save some time in data collections. However, these require the permissions from participants to access their data and use their record for secondary analysis. In conclusions, chromosome 3p2131 locus involved in COVID-19 susceptibility to severe respiratory failure, and blood group A individuals has higher risk, but blood group O individual has lower risk of susceptibility to severe COVID-19. Although this research has some limitations, improvement can be taken to allow further exploration of current findings. That's all of my presentations. Thank you.